I hope you are doing good. So this is teacher Neeta with the second online lesson. And yesterday we did introduction, general introduction, okay, encapsulation and blood. So we'll continue the same topic. It's a sort of assumption. Okay, we'll resume the same topic, but this time we're gonna do the platelets, okay, and blood platelets, okay, and the types of WBCs, okay. So the chapter name is same, circulation, okay, and this is our second lesson. I hope like you have gone through the first lesson, and if there is any question, okay, you can write me, okay, you can send your questions in Fedena, okay, or if you have my personal number, you can send your questions, okay, if you're not understanding anything, okay, I'll try my best to solve your queries, okay. So without much delay, let's start our second lesson. But before that, let me recapitulate quickly what we did yesterday. We did blood, okay, general characteristics of blood, then composition of blood, okay, the components of bloods when, plasma, RBCs, WBCs, blood platelets, okay, then the comparison between the, all these, right, in the form of a chart. I gave you chart for quick recapitulation, okay, and we just left with, I think, the types, okay, and detailed description of blood platelets, okay. So we'll start with the blood platelets today, right? So let's move towards our first slide, okay. So like this is actually the cellular part of blood, okay, blood platelets. Means we have RBCs, we have WBCs, and we have blood platelets, okay. So blood platelets are also called thrombocytes, okay, you already know. The other name, okay, biological name of blood platelets, okay, is thrombocytes, okay. Then next characteristic, blood platelets are not cells actually. Like, we studied WBCs, okay. We are saying like, it is coming under cellular part, okay, but these are not exact cells, okay. They are the fragments of the large cells, okay. So this is very much important thing, okay. That's why they are very small in size, okay. So what happened? Nucleus is absent here, okay, and the shape, if you're talking about the shape, okay, like we studied yesterday, the shape of WBCs, okay, RBCs, and here we are talking about the shape of blood platelets, okay. So the shape is random, no particular shape, okay. You can see in the diagram, okay, random, like you can say amoeba, like, structure, shape, right? So next is the size. The size is around 2 to 4 micron meter, okay. So micron meter, okay. If we are converting micron into meters, so it would be like it's around 10 raised to power -6, okay, meter. 1 micron meter is equals to 10 raised to power -6 meter, okay. So this is actually the size. So size is very small, you can say. And normal platelet count, okay, in the body of a normal person, like I said yesterday, is around 200 thousand, okay, but number varies, okay, but I think it's better to say 200,000 to 400,000, okay, per cubic millimeter of the blood. I mean, we are taking per drop of the blood, okay. So you can write, okay, like the number is not exact number, so you can write, okay, because it varies, okay, from person to person, okay. So it's just approximate value, okay. So these are some characteristics of blood platelets, okay. Next we'll do like the same thing, but quick recap, okay, what we did. Size is very small, okay, and granulated means granules are present in the cytoplasm of these blood platelets, okay, right? Small granules means small particles in the cytoplasm, okay. Then non-nucleated, okay. Then we can say but the maturation, on at the time of maturation, okay, we are talking about nucleus and non-nucleated and nucleated, okay. Sometime what happened at the time of just we can say in the beginning, okay, nucleus is not there, okay, but after maturation, okay, it's non-nucleated, okay. Then we have round or oval structure, okay, shape, okay. but it's not exact round or oval, okay, because the shape is irregular, okay, so it's better to write irregular shape, okay. So size is around 2 to 4 micrometer, and the number, you can simply write number is around 200,000 to 400,000, okay. It's from higher to lower, okay, but I think it's better to write the number from the previous slide, 200,000 to 400,000, okay, per cubic millimeter of the blood. And the lifespan is around 8 days, okay. This is the lifespan, okay. It can survive, okay, until 8 days, okay, right. So after 8 days, okay, there is actually the formation of new blood platelets, okay. So this is something, you know, about platelets. And then here is the structure and the composition, okay. Even though we don't have a structure, and structure we have, but not in detail. Detailed structure is not mentioned in our curriculum, but like we are doing online, so it's very difficult to make you people understand, okay. So that's why a small diagram is there. So I'll try my best to just give you something, okay, deep about these blood platelets, okay. So simple structure you can see, okay. I made this PPT in the form of very short, you know, paragraphs, okay, short, you can say sentences, okay. You can explain, okay, in your own words when you will write in the exam, okay. So blood platelets, you can say small plate-like structures, okay, and they're also known as thrombocytes, okay. What is the meaning of thrombocytes? Thrombo mean clot, like a small clot, okay, clotting of blood we are saying, and sites are cell, you know this thing, okay. And then we have size. The size is small, okay, the smallest blood cell, okay. This is very important point, okay. When you will you know, pen down, okay, you have to underline this point, okay, because if we are comparing the size of these blood platelets, okay, with size of RBCs and WBCs, the size is very small, okay. Like I said, it's around 2 to 4 micrometer, okay. And again, I'm repeating, you know, 1 micrometer is equals to 10 raised to power minus 6 meters. So you can like imagine, okay, the size. So then next is the shape, okay. Shape is like, you know, like shape is not regular shape, okay. You can say not particular shape, okay, but here the shape which is mentioned is spherical or oval or discoid in the form of a disc, okay. But when we are taking this blood platelets, okay, because blood platelets are formed from the larger cell, okay, so they are actually the fragments, okay. So that's why sometimes the shape is okay and sometimes the shape is not regular, okay. So volume, I said, okay, no need to write the volume, just write the count, okay. Just exclude this point, okay. The count is around 200,000 to 400,000 per cubic millimeter of blood. And then if we talk about the color is, you know, transparent, okay. Blood platelets are colorless, right. So that's all about the blood platelets, okay. I mean, general structure, a little deep structure, okay, the function, right. So let me repeat again. The function is, you know, the main function of blood platelets is clotting, okay, like as the name suggests, thrombocytes, okay, the formation of you know, clots inside the blood. Sometimes, you know, what happened if we have some sort of injury, okay, so the clotting of blood is happening, okay, because of these blood platelets, okay. So this is very important function, all right. So next, okay, we are done with the platelets, okay. So our next topic is types of WBCs, okay. Well, we did WBCs, that is white blood cell, yesterday, also known as leukocytes. But here our today's topic is, we did yesterday the characteristics, okay, the function, structure, count, everything, okay. But after that, you know, I left this part, okay. This part is a little detailed part, okay. So yesterday I left, so we'll begin today. The types, okay, because white blood cells are further divided into different, you can say, types, okay, subcategories, okay, on the basis of granules, granular and granular. Granules, again, I'm telling you, these are actually the small particles, okay, in the cytoplasm, okay. So we have 5 different types. We have lymphocytes, monocytes, basophils, neutrophils, and eosinophils, okay. So the percentage is around, the percentage of lymphocyte is around 20 to 25%, okay. Monocyte is 3 to 8%, like approximate percentage, and basophil is around 0.5 to 1%, you can say less than 1%. Neutrophil is around 60 to 70%, and eosinophils is around 2 to 4%. Okay, so you have to memorize the percentage because I'm going to ask you in MCQs, right? In first term exams are in your teacher exams, okay, in the form of MCQs or filling the blanks, okay? These things are important. So whenever you are studying, just make notes, all right, of all these small, MCQ things or fill-ups, okay? So next, the same thing, okay, but why I repeated the slide, okay, because here I'm just showing you the monocyte, okay. The monocyte is the first, we can say, beginning of you know one type, okay. It's a small subcategory of the WBC, okay, but after, you can say, engulfing some stuff, okay, after, you can say, because the main function of WBC is fighting, okay, because it is providing immunity, so it plays a very important role in the defense mechanism, okay. So what happened after, you can say, after encounter with some sort of antigens, okay, this monocyte is converted into macrophage, okay. A macrophage means little big cell, okay. So I was just showing you this, okay. So monocyte is converted into macrophage after, encounter with any sort of antigen. Antigen means any foreign agent, maybe bacteria, virus, or anything, okay. Now, next, we have types, we have function, and we have differential percentage, okay, like we did. But here, just quick, you can say, recapitulation. So first one, we have neutrophil. What is the function of neutrophil? The function is general phagocytosis, okay, acute bacterial infections, okay. Now, what is this phagocytosis, okay? I have a diagram, okay. In the next slide, I'll explain you with the help of diagram, okay. So its function is phagocytosis, okay. Then its percentage is around 54% to 62, like, I think it contribute the major part, right? Then we have eosinophils, okay, it kills the parasite, and allergic conditions are because of this, okay. So its number is around, percentage is around 1 to 3%. And then we have basophil. The function of basophil is to release heparin and histamine, okay. Now, what is this heparin, okay? Let me first explain you just tell you the percentage. Percentage is 1%, okay. And go back to the point, heparin and histamine, okay. So heparin is actually anticoagulant, okay. Now, what is coagulation? Coagulation is again the same thing like clotting, okay. Another word for clotting is called coagulation, but this is actually something anticoagulant, okay, or we can say this is a blood thinner, okay. Now, what is the meaning of this? Because sometimes what happened, there is, excess clotting, okay. We have clots inside our body sometime, okay, because of some infections, because of some problems, okay. Clots are formed inside our body. So basophil, is playing a very important role here, okay, to release heparin, okay, in order to clear those clots, okay, dissolve those clots, okay. Next, we have histamine, okay. So histamine is also actually a chemical, okay, released from WBCs, okay. But WBCs, we have different types of WBCs, like 5 types, okay. But these 5 types are further divided into different subcategories, okay. So we'll not go into detail, okay. So, but I'm just telling you, histamine is released by specific cells, okay, and these cells are known as mast cells, okay. So it is actually a chemical, okay, and it actually it is released, okay, in response to some sort of allergens, okay. So again, its function is to fight, okay, and provide immunity, okay. So I hope you got what is heparin and what is histamine, okay. These are the chemicals, okay. One is anticoagulant, okay, means dissolve the clots, and other one is actually you can say defensive in mechanism, okay, right. And its percentage is around less than 1%, okay. Then we have monocyte. Monocyte, the function of monocyte is phagocytosis of the large particles in case of your typhoid or malaria, okay. So what is this phagocytosis? I'm going to explain you, okay. Just leave this for now. So its percentage is around 3 to 9%, okay. Then we have lymphocyte. Okay, we have B lymphocyte, we have T lymphocytes. Okay, we have many types, but we are not going into detail. And its function is to provide antibodies. Okay, and its percentage is around 25 to 33%. Okay, so I hope you understood. Okay, but in the next slide, I'm explaining you what is phagocytosis. Okay, phago is basically engulfing. Okay, like eating something. All right. Okay, cytosis means cell. Okay, like inside the cell, this is actually a process, okay, right, performed by cell. In this process, what happened, like you see, we this is actually the cell, all right, okay. But when this cell, okay, comes in contact with some sort of bacteria or viruses or any foreign agent, okay, the cells, the cell wall, okay, basically the cell membrane, because we are talking about animal cell. So the animal cell membrane, okay, has special type of receptors, okay. So after binding with these receptors, cell try to engulf this, okay. So like you see here, receptors, okay, these are receptor proteins, and bacteria, okay, the red, tablet shaped structure, okay. So after this, what happened? Bacteria engulfed by the cell, okay, and now the bacteria is inside the cell, okay, and it forms a structure known as phagosome, okay, after phagocytosis, okay. The structure is called phagosome. Basically, this is a bacteria, okay, and some receptor proteins, okay. And next to this, we have lysosome inside the cell, lysosome, suicidal bag of the cell, right? So lysosomes contain around 40 different types of enzymes, okay, in order to dissolve the foreign agents, debris, dead things inside the cell, okay. So now we have this phagosome, okay, and inside, like basically phagosome is bacteria, okay, right, covered with some, receptor proteins and other stuff, right? So what happened here? Now this phagosome and lysosome, okay, this phagosome is here. Now we have lysosome. Phagosome and lysosome combine together, okay, right, because we need enzymes to dissolve this bacteria, okay, and together they form phagolysosome, okay. So after the formation of this structure, what happened, the lysosome contain 40 different types of enzymes, like I said, okay. So all these enzymes are actually dissolving this bacteria, the foreign agent, okay, like you saw here. It is fragmented into 4 parts, then many parts, okay. So this is actually the last stage, okay. After this, what happened? Now bacteria is dead, okay, cell is free from any sort of infection, okay. So what happened? The dead debris or whatever is there, okay, which is of no use, okay, and it's now not harmful, okay, but cell is actually throwing it out of the body, right? So this process is known as exocytosis, all right. So first, phagocytosis means how cell is actually responding to the foreign agents, okay, right? They actually, cell is engulfing, okay, and killing that foreign agent. I hope you understood what is phagocytosis, okay, because in the last slide, okay, the function of neutrophil and monocyte is related to phagocytosis, okay. So I thought like it's very important thing, so I explained you with the help of diagram, okay. Now the next, we have all the types, okay, and actually these are the subtypes of WBCs. Then we have nucleus, okay. So we will actually you can say explain, right? Okay, we'll discuss everything, all right. The structure is here, the function is here, the nucleus type, okay, and subtypes, okay. So what type of nucleus is present in neutrophils, basophils, okay, and what is the function, and with the help of example, or we can say this is structure, right? So let's begin with the first one, which is neutrophil, okay. So if you will see the structure inside this, the nucleus is multi-lobed, okay. Multi-lobes mean like many bulges are there, okay, right? 1, 2, 3, 4, 4, 5, I can see, right? So what is the function of neutrophils? Okay, the function is bacterial or fungal infection, okay. And actually these are the most common first responders to the microbial infection, okay. So you can say like the function is, what is the function of these neutrophils, okay. These are the first responders, because the number of neutrophils is more as compared to the other, you can say, subtypes, okay, right? So what happened here, this is the function, okay. Next we have eosinophils, okay. So the structure, if you're seeing the structure, second diagram, okay, Inside this, what happened? We have nucleus, and nucleus is bilobe structure, okay? And it is, what is the function of this eosinophil? Okay, it is parasitic. You know what happened? Parasitic infections and allergic reaction, okay? Like basically it is related to inflammation, okay, right? When something is happening, okay, inflammation is caused, okay, are performed by these eosinophils, right? Now, next we have basophils, okay? So the nucleus inside this is white, trilobed, it depends, okay, right? Here you can see it's not like you know, rigid thing. It can be white, can be tri, okay? Now, next we have the function, these basophils, okay, the function of basophil is allergic and antigen response, okay? The same thing I'm saying like we did, it release actually, it releases, you know, histamine, okay, and causing the vasodilation, okay? Now, histamine, you know, okay, histamine is basically the chemical, okay, released by the basophils, okay, but further we have types under the basophils, and that type is mast cell. So it causes the vasodilation, okay? Now, what is vasodilation? Vasodilation is actually the widening of the blood vessel, okay, in order to supply more oxygen, okay? Means more blood, okay, because blood contain more oxygen, okay? So where, like, you know, cell, so for example, you know, if there is a scarcity of blood, okay, so in that case, what happened? Basophils, okay, because of any allergic reaction, okay, because of any allergic thing, antigen, okay, bacteria or virus, okay, so in that case, what happened? Okay, basophil is giving a signal, okay, signal to these mast cells to release the histamine, okay, and histamine cause the vasodilation, okay, means supply more blood over there, right? Now, next we have lymphocyte, okay. Now, lymphocyte, okay, if we talk about the nucleus, okay, so nucleus is deep staining, like, you know, stain is basically the dye, right? Okay, so the nucleus is deeply stained means because it's darkly stained, okay. Then it is eccentric, okay. So eccentric, you know, eccentric, concentric is different, okay. This is actually eccentric. What is the meaning of this? Means there is no center, all right, okay, it is not occupying a particular center. You can see, you know, one edge is here, okay, it is touching the boundary, and another one is not touching the boundary, okay. So this is the meaning of, you know, eccentric, okay. But we have concentric. Concentric is different thing in mathematics, you guys have read, I guess. It is you know, something, if one, you know, two these you can say figures are sharing the same center, okay, so I guess that thing is called concentric, okay. So eccentric is basically without any center. So here you can see there is no center, right? Next we have the functions, okay, the functions of these lymphocytes, okay. So it includes the B cells, okay, CD4 plus helper cells, T cells, and CD8 plus cytotoxic T cells. Basically these are the types of lymphocytes, we can say, right? Okay, all these are the types. We have B type, C type, okay, we have T types, okay. So don't worry about all these types, okay. You can simply say it operates primarily in the lymphatic system. This is main cause, okay, main you know, function of your lymphocytes, okay. Next we have monocytes, okay. You can see the diagram, okay. The nucleus is basically kidney shape, okay. You can check bean shape here also, kidney shape, somewhat kidney, not exact, right? Okay, but we are actually just comparing, okay. And what is the function? Its function is phagocytosis of the pathogens. The same thing we did, okay. What is phagocytosis? You understood now. And presentation of the antigen to T cells, okay, because I mean, it is taking actually the antigens to T cells, and eventually they become tissue macrophage, like I showed you in the third or fourth slide, okay, how, you know, monocytes are converted into macrophage, okay, which removes the dead cell debris and attack, you know, microorganisms. So what happened? This is the main function, okay. Means, but the main thing is here, you know, the function is phagocytosis, the same thing, okay. But after engulfing those infected things like bacteria, the monocytes are converted into macrophage, okay. So I hope you understood these types, okay, functions, right, with the help of you know, structure, okay. I gave some examples basically the structure only, right? And but we categorize, you know, we divided on the basis of nucleus, function, and examples, okay. So these are actually the types of WBCs, okay. Next we have another very quick topic, okay. hemoglobin, right? Okay. So what is hemoglobin? You know this thing, okay? Is this exclusively found in case of RBCs, okay, red blood cells, and its function is to transport oxygen and taking out carbon dioxide from the tissues. I mean, giving oxygen to the tissues and taking out the waste materials from the tissues. Okay, and we have different types of hemoglobins in our body, right? But the most common one is hemoglobin type A. Okay, and it's composed of 4 polypeptide chains. Okay, polypeptides mean made up of many. Poly mean many. Peptide means made up of peptide stuff. Okay, and peptide is made up of amino acids. Okay, right? So it is composed of 4 polypeptide chains. Okay, 2 alpha and 2 beta. Okay, held together with a non-covalent interaction. Okay, right? I'll show you the structure. Its structure is mentioned over there. And next, we have each day 6 to 7 grams of hemoglobin is synthesized to replace the loss through the normal turnover of RBCs. So 6 to 7 grams is synthesized every day. Okay, this is very important thing. Okay, and the last one is each subunit has stretches of alpha helical structure and a heme binding. Okay, pocket. So this is a very deep structure. Okay, but you can see in the structure, right, what I'm talking about. Here you can check. Okay, the heme group. Okay, here like the flower like structure. Okay, these are the heme groups. Okay, and we have two chains. Basically this is actually the alpha alpha. Okay, and here we have beta beta. Okay, right? So like I said in the last slide, okay, is composed of the 4 polypeptide chains. Okay, so you can see here 1 2 3 4, 2 alpha and 2 beta, right? And in the center of these chains we have heme group, right? That's why it is called hemoglobin, right? uh, but that was actually the complicator, I mean the real chemical structure. Okay, and this is very simplified structure. Okay, like you can just check here the round globulin chain. Okay, right? And globulin chain is at the boundary. Okay, and heme group is in the center. Okay, so this is the simplified structure, just to make you guys understand that what is actually the structure of hemoglobin, a simplified one. Okay, so the normal value of hemoglobin, okay, in case of you know humans, like male, female, children with the different age groups. Okay, right? So what happened at birth? Okay, like a baby, right? Baby is having around 13.5 to 19.5, okay, gram per deciliter of this hemoglobin, right? Okay, now what is this DL? DL is deciliter, like I said. Okay, deciliter is also a unit. Okay, deciliter is a unit to measure the hemoglobin value, right? So what is the value of actual deciliter? Okay, like I said, I told you the value of micrometer. Okay, in the same way the value of deciliter is 1 deciliter is equals to 10 raised to power minus 10 liter. Okay, this is actually very important thing. So now you can imagine, right? So children with the age group 2 to 5 years, sorry, you can say the amount of hemoglobin in these children, okay, with the age group 2 to 5 years, okay, is 11 to 14 grams per deciliter. Okay, and children with 6 to 12 years, okay, is around 11.5 to 15.5, okay, gram per deciliter. And the last two things are very important. In the case of adult female, okay, its value is 13 to 15 gram per deciliter, and in case of male the value is 14 to 16 grams per deciliter, right? So like these values are approximate values, okay, not rigid, but the approximate values. Okay, so you must know these values, okay, because we are studying the circulation in detail. Okay, and you guys will face many competitive exams after your grade 12, right? So just keep all these things in mind. Okay, so we are done, okay, with our today's topic. Okay, just a quick recapitulation in 2 minutes. Okay, what we did blood platelets. Okay, because yesterday we left this topic. Then we did structure of the blood platelets. Okay, then types of white blood cells. Okay, then functions, then phagocytosis, which is very important. Okay, then comparison, okay, of all these 5 types. Then next topic was the hemoglobin, okay, and the structure of hemoglobin and the normal hemoglobin value in case of children, adult, okay, and a baby, right? So I hope you understood, okay. If you have any question, okay, you can write me, okay, you can ask me. You can write me in Fadina, okay. You can send your question, or you can, if you have my number, okay, you can write me on WhatsApp, okay. I will try my best to answer your questions, okay. So that's all for today. Thank you so much. See you soon in the next lecture, okay. Bye bye. Take care.